Imbibers, a very good afternoon to all of you. Once again, we are here to discuss the another topic from the molecular basis of inheritance, which is the RNA world. Yes, we have discussed about, we have given you the introduction towards the DNA, and today we're going to discuss about the RNA. What is RNA? What kind of a structure does the RNA have? What are the types of RNA is there? Yes, I know we're still, the next session we're going to basically discuss about the DNA packaging and everything. Yes, but I wanted to give you a little introduction about the RNA also. Before, we have decided already that DNA is a genetic material, why RNA is not a genetic material. So it is important for you to learn about why the RNA was not the genetic material and what is the structure of RNA. So without wasting any time, let me introduce you to myself. I am Anam Khan. I'll be your botany expert for the session 2023 and 2024, and we're going to discuss all the amazing and interesting topics related to biology. Yes, like we have already completed our one chapter, which is sexual reproduction in flowering plants, and we have done lots of practice around that particular chapter. If you have missed any session, the session is already on your dashboard, on the Embibe dashboard. Whenever you have time, you can go through them. Yes, so please join us on the Embibe. All the upcoming classes have been scheduled for you so that you can be easily register yourself for the upcoming classes so that you never miss an update. And there's always a pre and the post class activity before you join the class. Yes, the pre-class activity is for you so that you know what is going to be taught in the class, so you come a little bit prepared with the questions and your queries. On the other hand, there is a post-Embibe classes that after the class is done, you're going to attempt the post-Embibe classes to know that whether you have understood the concept or not. Yes, so without wasting any time, let's start our today's session, which is the RNA world. Yes, it is one of the topics from the molecular basis of inheritance. Talking about the RNA world, yes, this chapter, which is the molecular basis of inheritance, the topics to be covered, we have already discussed about the search of genetic material. Yes, where we have discussed about the respective experiment performed by different scientists to prove that DNA was a genetic material, and also why the RNA was discarded as a genetic material. Next, we have discussed about the DNA structure. We are still going to discuss about the packaging after we discuss about the RNA world. After that, we're going to start with the replication, transcription, genetic code, translation, regulation of gene expression, human genome project, and DNA fingerprinting. All of these things we're going to basically start in the coming sessions. Yes, let's see what we have today. In today's chapter, today's session, we're going to discuss only about two things, two concepts to be covered. One is the RNA, the other is the structure of RNA. These two concepts are to be covered. This is the RNA world, and these two concepts have to be covered in this particular session. So before we start our today's session, let's have a quick recap of what we have studied in the previous sessions related to the DNAs. Yes, we have discussed about the DNA, the length of the DNA. We have discussed about the structure of the DNA, the nucleotide, the nucleoside in detail about all. If you have missed any session, you can already go and watch in the Embibe app. What is this? Yes, can you see this? Do all three models belong to a single macromolecule? Do you think that this model, and this model all belong to a one single macromolecule? Yes or no? What is the answer? No, the structures are different. As I can see, the structures are different. They all do not belong to a one single macromolecule. Yes, they are basically belonging to the two different macromolecule, the DNA as well as the RNA. Yes, talking more about the RNA, we have very first thing talking about the RNA, the basic introduction, the basic features of the RNA, that RNA is the first, it was actually thought that it was the first genetic material because that is served as a basis of evolution of a key life system as metabolism, translation, splicing and others. So children, initially in the initial years of research, in the initial years of experiment, it was thought that RNA was the genetic material. That is what we have already discussed, that why RNA was thought to be a genetic material. The reason that it shows the basis of the translation, the key life system as a metabolism. Yes, and the translation because it was creating proteins. Yes, and you know that proteins are the building blocks. Yes, all of you know that the proteins are the building block, the splicing and the other. It played a major role in the evolution of DNA. Why it has played a major role in the evolution of DNA? If you see the basic process that DNA will create what? mRNA. Yes, and the mRNA will create proteins. Yes. So whatever changes will be happening in the DNA is going to reflect in your body through this mechanism only. This mechanism from the protein synthesis, because you know that the proteins are the building blocks. So whenever there is going to be an evolution, there is going to be a change in the genetic material. This genetic material in the particular organism will show their changes, their evolution process will be shown through this only. Yes, because all the things are interdependent on each other. So that is why in the initial years it was thought that RNA was the genetic material, the basic of the evolution process, the key life system such as metabolism, translation, splicing. All these things we're going to study in future. Yes, what is splicing also, we're going to see what is translation also. But since we are only giving the introduction towards the RNA, you know that RNA undergo the process of translation to make proteins, undergo the process of splicing, eradicating the non-coding sequences and also the other metabolic activities. Yes, it plays a major role in the process of evolution. Yes, the whole soul changes in the DNA would reflect through the RNA only. Yes, so this is the introduction towards the RNA, which was initially thought to be the genetic material, but later on it was discarded and thought that DNA was the genetic material from the experiments done by the different scientists. Talking about the characteristics of the DNA, the genetic material, if I talk about children, the genetic material in many ways, viruses like retroviruses and plant viruses. Yes, so genetic material, talking about, as you can see also, characteristics of the DNA, characteristics of the RNA. RNA is a genetic material of several viruses. Many viruses like retroviruses, plant viruses serve as a genetic, their basic genetic material is only RNA. There is no existence of the DNA only. They have a genetic material, they have their information stored in the form of RNA only. So the RNA serves as a genetic material in some viruses. Yes, some viruses like retroviruses and some viruses like plant viruses. Yes, it catalyzes important biochemical reactions also. Yes, the RNA catalyzes the important biochemical reactions which are taking process in the building. Yes, it catalyzes that particular process. Simple. What are the biochemical reactions of basically creating the proteins also? Yes, it is basically catalyzing the whole process of the creation of the proteins through the mRNA machinery. Yes, and they are very reactive and unstable. Yes, all of you know about the COVID-19, which is a virus which has a genetic material RNA. It was able to cause infection in every particular, globally it has called infection because it was able to mutate faster. It was able to mutate at a very faster rate. And the reason it was able to mutate at a faster rate because it has a genetic material as a RNA. Yes, the genetic material in the particular viruses was RNA, and RNA is very unstable. They evolve very, I would say, at a very faster rate. And when they evolve very faster rate, they are basically going to survive under any. They might not be controlled by several medications also. Yes, there was several pandemic because of the virus only, which has a genetic material as an RNA. It is completely very reactive and it was unstable as well. Yes, so the RNA is absolutely unstable and very reactive. Due to this particular feature also, it was not considered as a genetic material globally. Yes, it was not. There might be certain viruses having the RNA as a genetic material. There might be plant viruses which might be considered as a genetic material as RNA, but you know that basically we always know, since the DNA is less reactive and it is more stable, and that is the reason we have actually confirmed, the scientists have confirmed through their various experiments that DNA was the genetic material, because the RNA was absolutely unstable and very reactive, and in turn the DNA was very stable and less reactive. Yes, so these are the characteristics of the RNA. I hope that is clear to you. Yes. Clear? So this is about the RNA. Moving forward, talking more about the RNA, next we have the structure. What is the difference between the structure of the RNA as well as the DNA? Talking about the RNA, children, it is not a double stranded structure, it is single stranded structure. Yes, it is a single stranded structure. The ribose sugar. Yes, talking about the ribose, talking about the structure of RNA, it is a single stranded and it is made up of mainly ribonucleotide. Yes, the nucleotide is having a ribose sugar, it is called ribonucleotide. The components present in the structure of the RNA is a ribose sugar, nitrogenous base and phosphate sugar. In the particular DNA, we have a sugar called deoxyribose sugar. Yes, we have a sugar called deoxyribose sugars. Why the ribose sugars are present in the DNA? Yes, we have a nitrogenous bases, we have a phosphate group. Yes, this is the structure of RNA. It has a phosphate group, same as DNA. It has same basic structure as DNA, other than the whole sugar is different from the DNA, which is a deoxyribose sugar, and here we have a ribose sugar. In the DNA, we have deoxyribose sugar. Other than all the things are similar. There is a phosphate group, there is a nitrogenous bases, yes, purines and pyrimidines, and instead of thymine, there is a uracil. Yes, in the nitrogenous bases, there is A will be combining with the U, which is a uracil. G will be combining with the cytosine. There will be uracil instead of thymine. Yes. Next, talking more about the structure of RNA, talking about the RNAs as a ribose sugar. If you see the structure, yes, I always talk about the series structure. This is, if I say this is 1 carbon, 2 carbon, 3, 4, 5. Yes, this is a 5 carbon compound. It is a cyclic structure containing 5 carbons. Why this is more reactive? This is what we know. It is comprising 5 carbons, 10 hydrogens and 5 oxygen molecule. Yes, this 1, 2, 3, 4, 5 oxygen molecule, 2, 3, 4, 5, 6, 7, 8, 9, 10. 10 hydrogen molecules. Yes. Talking about the structure of the ribose sugar, we have a cyclic structure which having 5 carbon, 10 hydrogens and 5 oxygen molecule. The second group of carbon in the ribose sugar is a very chemically reactive hydroxyl group linked to it. This is what we have also, I have also told you in the DNA, when we are discussing about the DNA, that there is a presence of oxygen, there is a presence of hydroxyl group at the second carbon, which makes the particular RNA very unstable and very reactive. Yes, because it is very unstable and very reactive due to the presence of hydroxyl group at the second carbon, because it is linked to the second carbon, which makes it very susceptible to the hydrolysis. Yes, because of the OH group at the second carbon, it is very susceptible to the hydrolysis process. Okay, because the DNA does not have a very reactive OH group in the same place as RNA. Yes, there is a deoxyribose sugar. Deoxy means there is a deletion, the subtraction, there is an absence, whatever you wanted to say, you can say there is an absence of oxygen atom at the second carbon in the DNA, and that is why it makes us less reactive. there's a less reactive nature of the deoxyribose nucleic acid, which is a DNA, is because the absence of oxygen group at the second carbon, which makes it more stable and favored as a genetic information in the most creature, because we do not want that evolution process might take 100 years, 10,000 years in the process of evolution, but we need a particular genetic material which is more stable, and in the coming particular generation it shows stability. Yes, the children, that is why, because the DNA was less reactive, due to this more stable, that is the whole reason that we wanted the particular DNA as the genetic material. It was more stable and it was the most important component for information transfer in the most creatures. Yes, and it has bonded with the nitrogenous bases and the phosphate group to form ribonucleotide. Adjacent to a ribonucleotide, bases are chemically attached via phosphodiester bond. Yes, exactly same as you can see, it is bond with the nitrogenous base and a group of forming a ribonucleotide, and basically the nitrogenous base is attached to the phosphate group and they have form a ribonucleotide chain, and then adjacent to ribonucleotide bases chemically attached in a chain via phosphodiester bond. Yes, they are being attached to the phosphodiester bond. Since they are not similar to the double stranded, it is more of a single stranded, and this is how the structure of the ribose sugar is formed. Yes, I hope this is clear. Why the ribose sugar? What is the structure of ribose sugar? Yes, adjacent to ribonucleotide bases only, chemically attached in a chain via phosphodiester bond. The whole chain of a ribonucleotide is attached via phosphodiester bond. Yes, this is about the RNA as a ribose sugar. Next we have the nitrogenous bases in the RNA. Very simple, talking about the nitrogenous bases. We have as I have already told you, we have adenine, guanine, cytosine and uracil. Uracil instead of thymine. Thymine is present in DNA. It is a part of a DNA. So, the presence of uracil also in the RNA makes it more reactive and unstable. And in simpler words, when you talk about the bearing of the purines and pyrimidines, the adenine paired with the uracil, A, T with the 2 hydrogen bonds, and the guanine pairs with the cytosine with the 3 hydrogen bonds. This is, we have already talked about. The thymine, sorry, I should place U, sorry. So, talking about the RNA, A, T will be replaced with the uracil with the 2 hydrogen bonds, while the guanine is going to pair with the cytosine with the same 3 hydrogen bonds. So, this is the nitrogenous bases. This is the whole structure of the nitrogenous bases involved in the grouping of pyrimines and pyrimidines. Same 2 bonds will be present between AU, and GC will have 3 hydrogen bonds between them. Yes, I hope this is clear. Yes, talking about the nitrogenous bases. Next we have a phosphate group is present at the 5 end of each nucleotide. Yes, at the end of each nucleotide there is going to be a phosphate group. Yes, to form a whole particular chain of a ribonucleotide, there is going to be a phosphate group, and each nucleotide will be joined through the phosphodiester bond. This is what we have already discussed, but these phosphate group will be present at 5 end of each nucleotide group. 5 end. Yes, it will be always at the 5th carbon. There is a phosphate group will be always attached in the nucleotide. Yes. Talking more about the RNA, next we have the comparison between the DNA as well as the RNA. What is the comparison between the DNA and RNA? What are the features which make them not similar to each other, and basically their characteristics also differ from each other? Talking about a DNA, it is a chemically a nucleic acid. Same RNA, it is chemically a nucleic acid. Both are nucleic acid. DNA and RNA both are the nucleic acid. Yes. DNA is a chemically a nucleic acid, where the RNA is also chemically a nucleic acid. Genetic material in prokaryotes, eukaryotes and some viruses. Yes, majorly the genetic material in various prokaryotes, eukaryotes and some viruses is the DNA. Genetic material in plant is viruses and retroviruses. Simply, RNA does not being a genetic material in most of the thing, but the viruses generally have RNA as a genetic material. The particular DNA will be double stranded while the RNA is mostly single stranded. Yes, the sugar is absolutely which makes them a very different nature, chemical nature from each other, is that it is has a deoxyribose sugar while the RNA will be having a ribose sugar. The DNA will be highly stable while the RNA is less stable. Yes, nitrogenous base are adenine, guanine, thymine and cytosine. Talking about here, the nitrogenous bases are adenine, guanine, uracil and cytosine. There is a uracil instead of cytosine. It cannot work as a catalyst, but RNA can work as a catalyst. So children, these are the major characteristic difference between the RNA and the DNA. I would say, since the particular characteristics of DNA being less reactive and more stable makes it a genuine genetic material, it is a source of transferring information from one generation to another. On the other hand, since RNA is basically more reactive and highly unstable due to the two things, because of the uracil also and because of the presence of hydroxyl group at the second carbon. It also works as a catalyst because it is very reactive. Similar, the nitrogenous bases are different in case of urine, pyrimidine and pyrimidine, and the bonding is also different. The AT, thymine, will instead of thymine in the RNA, there will be a uracil bonded with the 2 hydrogen bonds, while the guanine will be bonded with the 3 hydrogen bonds with the cytosine. This is the major difference between the RNA as well as the DNA. Next, we have a question for you children. Yes, which of the following is not a characteristics of an RNA molecule? What do you think is not a characteristic of a RNA molecule? Yes, what do you think is a characteristics of an RNA molecule? Highly reactive, adenine and guanine is present. It is a nucleic acid genetic material in all the viruses. What do you think is a characteristic of an RNA molecule? What is the correct answer? Which of the following is not a characteristics of the RNA? Just now we have discussed all the characteristics of the RNA. Highly reactive, adenine and guanine is present. It's a nucleic acid and it's a genetic material of all particular what viruses? Yes or no? The correct answer is genetic material in all viruses. No, it's not true. Some viruses do also have DNA as a genetic material. Retroviruses and especially plant viruses are majorly having RNA as a genetic material. Very simple. Next, we have another question for you. Which of these nitrogenous bases are unique to the RNA? Yes. Which of these nitrogenous bases are unique to the RNA? What do you think is unique to the RNA? Adenine, thymine, cytosine, uracil. Which among them is unique to the RNA? Yes, which is present in the DNA but absent in the RNA, and some which is present in the RNA and absent in the DNA. What could be the answer? Yes. Correct answer is the uracil. Uracil is there present in the RNA bonding with the adenine. Yes, instead of thymine, there would be a uracil in the RNA. Next question. Which of the following is an example of nucleic acid? Yes. What do you think is an example of a nucleic acid among them? Yes. Which of the following is an example of the nucleic acid? DNA, RNA, both A and B, none of the above. Correct answer is both A and B. The RNA and DNA both are the example of the nucleic acid. Next we have, so coming into our particular session, we have discussion about the RNA. What we have discussed about that RNA is a first genetic material. It was thought to be the first genetic material, and it serves as a basis of evolution in crucial life system as well as metabolism. Yes, translation splicing. Initially, it was thought to be the genetic material. The genetic material of various viruses like retrovirus, plant viruses is majorly the RNA. RNA catalyzes the important biochemical reaction, and it is very reactive and unstable. It is made up of ribonucleotide with ribose sugar, a nitrogenous base, and a phosphate group. The RNA ribose sugar has a cyclical structure comprising 5 carbons, 10 hydrogens, and 5 oxygen molecule. The second carbon group in the ribose sugar molecule has a chemically reactive hydroxyl group linked to it, which makes RNA susceptible to hydrolysis. Adenine pairs with the uracil 2 hydrogen bonds, guanine pairs with the cytosine and 3 hydrogen bonds, and the phosphate group is present at the 5 end to each nucleotide in RNA. All the things, all the related information of the RNA has been already shared with you. Yes, so children, what we do in the end, we have a quick, what does our DNA contain to be found newly formed cell? How does DNA continue to be found in the newly formed cell? What is the reason? Yes, we're going to discuss that in the next lecture. Next, this is what is your Embibe app. You can log in through your phone number and your email ID. As soon as you land on the home page, you will see the Embibe live classes. All the upcoming classes have been scheduled. This is the one-stop solution for everything. You can learn, you can practice, you can test, you can see how much you have achieved. And children, you can go through everything, through every solutions from your particular subjects. Next, we have the Embibe Lens app, where you can just scan the text. It will highlight everything in the every keyword and will show you everything in the 3D. Yes, so I really want you to be there. Of course, yes, this is the one-stop solution. This Embibe app is amazing. You can explore the Embibe app, and you can also join us for the practice session, so you can directly answer through the Embibe, which gives you an opportunity to directly answer via your computer, via your phones. Yes, which is not available at the YouTube during the practice session. Until then, we will meet again discussing about the packaging of the DNA in the next session. Until then, please take care, keep imbibing. Bye-bye.
Keep imbibing. We believe in you.